because when people are planning and doing conservation measures, they had to look at what ecosystems can give to human beings, especially that most of the humanity now lives in urban areas. So the take home is that planning should incorporate ecosystems, and in particular wetland ecosystems, because they are sensitive ecosystems and they are endangered. Two, they have a lot of biodiversity, and it is generally accepted by nature-based solutions in planning, and even intergovernmental and the UN bodies on the relationship between nature and science and the social and ecosystems within urban areas, is that biodiversity is the foundation or the basic unit that you need to consider when you're looking at ecosystem services. So the biodiversity of wetlands, therefore, in relation to that they are scattered within urban areas, is so critical for maintaining ecosystem services within urban areas, and therefore should be mainstreamed or considered in urban planning. For example, in the research that we did, we found out that some of the species that are in this wetland
entitled Effect of Dietary Inclusion Watering Jewweed on Growth and Bacterial Community Composition in Farmed Edible Crickets. In this case, I'm looking at the field cricket, the species called Gyrus bimaculatus, or field crickets. As an introduction, we all appreciate that by now that edible insects are highly nutritious, and among the commonly consumed insects, for instance, in East Africa, we know termite, we know grasshopper, we know black ants. Recently now we have crickets. But this practice of consumption of insects is actually associated with wild collection. They're actually collected from the natural environment. So in this perspective, one of the challenges, or some of the challenges we normally encounter, is they are not sustainable because they are only consumed when they are available. For instance, for grasshopper, and they are swarming. We have some issue with some food safety concern. We don't know what insects are feeding in the wild. We control the feed. We cannot control the feeds they are feeding in the wild. So we have some pathogenic and food safety concern. Also, another challenge is that because of their high water activity, they are highly perishable, and this is a challenge. Sometimes we have abundant harvest of grasshopper, but we don't have a way to preserve them. So what is the main focus currently? What are the researchers doing? The researchers are currently looking to domesticate them from the wild and then bring them to captivity. So we are doing sustainable farming in a controlled environment. And then the researchers are also doing called diet manipulations to try to enhance their nutritional profile. And something also very important is about the safety, very important. That one has to be guaranteed. Now, of course, in this area, I'll be looking at the safety. What are the food safety concerns? So the question everyone would ask, is it safe to eat insects? Of course, we have been eating insects, but there are people who are not accustomed to eating insects. So the question would be, is it safe? Some of the major hazards have been grouped into 3 categories. So we have the microbial hazards. So you have bacteria and fungi. You have chemical hazards. So in this case, you have metals, pesticide residues, and mycotoxins. And we have allergies. So, but for the purpose of this presentation, I'll concentrate much on the microbial hazards. So we are saying that the effect of dietary inclusion of watering jewweed in cricket farming. So why watering dew? We should appreciate that currently cricket farming has shown potential for large-scale production. There's a lot of research on cricket farming. Many people have actually adopted the cricket farming. But the challenge they have is that the high cost of local feed. For instance, most of the farmers I know, they use chicken feed, but they are quite expensive. You have to get these from the local dealers. So what is the main focus from this? So the researchers are trying to use locally available feed materials that are cheap and also locally available. For instance, a study by these two researchers, they tested 5 types of weeds that are commonly found in our farm weeds. These included the waddling dew weeds. Among those 5 weeds, waddling dew received the highest acceptability. Also, looking at the literature, waddling dew has also been shown to exhibit some antibacterial activities against some common foodborne pathogens. So I took it from them, this Kinyura and Koech, to look now the impact of including this waddling dew during the cricket clearing on different body growth parameters, that is body weight and mortality, and the microbial quality. The reason why I also look at the microbial quality is because of this point, they have been shown to exhibit antimicrobial activity against some common foodborne pathogens. So in simple, this was the setup on the methodology. Actually, this is the waddling dew for those people who don't know what I'm talking. It's very common, especially where you have maize plantations. So the experimental setup, the species was a Grylas immaculate or the field crickets. So in this case, I had 2 experimental treatments. I had the control, which is the starter chicken feed only, and then starter chicken feed supplemented with waddling dew. Then these were the feeding regime here, 2 gram per layering container per. Of course, each experimental treatment had 3 replicates. These are the feeding protocols. So the rearing duration was 30 days. These are the parameters that I actually monitored. I look at the body weight of the crickets and the mortality. I also look at the microbial counts. And later I also did the metagenetic analysis, the screening of microbial diversity that are associated with these crickets. Looking at the body weight and mortality, we observed that at the end of 30 days of layering, the crickets that fed starter chicken feed that was supplemented with waddling dew had significantly higher those that were fed on chicken feed only. So this one looked promising because that means you can actually supplement chicken feed. You are cutting on the cost, and you're also getting the cricket with the higher sellable weight. Looking at the mortality, on average, you found about 15%, but there were no significant difference between the 2 treatments. Look at the microbial counts. Of course, these are the major microbial counts. We are currently looking at the insect because right now we are looking insect as meat because of the high protein content. So we have adopted this count from the meat. We found that generally from the 2 treatments, both insects were highly above the recommended limit. But what was promising is that we found that, especially this case for the bacterial endospore, we found that those crickets that fed chicken feed that was supplemented with waddling dew had significantly lower count of bacterial endospore than those that fed on chicken feed only. And this is very important because remember, endospore, these are the main bacteria that are giving us challenge when it comes to the processing of crickets and cricket-based products, because these ones, they are not easily eliminated by heat processing methods. So we found that this one also promising. When we looked at the metagenetic analysis, this is actually the aim of metagenetic analysis is screening all microbial diversity that are associated with crickets. In general, we found that both crickets between the 2 treatments were actually dominated by the same bacterial community. But of course, the Coxella, which is a family, then the Lactobacillus and the Spiroplasma were the major bacterial community. Another important reason why we do metagenetic sequencing is that you'll be able to identify whether there are bacterial groups that are associated with foodborne safety or foodborne pathogen bacteria. In this case, for instance, we found there was Clostridia, which is considered like for Clostridium botulinum. We also found some Staphylococcus and of Enterobacteriaceae. But I'd like to emphasize that, remember what we have presented here, these are DNA sequences. So it doesn't tell us whether these bacteria they are alive. It doesn't tell us the nature of the bacteria, whether it's alive, dead, damaged, or cultural. So we just assume that this, for instance, this bacterial group is associated with this bacteria, but further tests need to be done to consider whether it's dead or alive. In conclusion, we found that from this short experiment, we found that actually we can tap waddling dew in insect feed formulation, and you can get crickets with high body weights instead of just giving them the chicken feed, which is quite expensive. We also found that we can have some possibility of reducing risk associated with bacterial endospores. Remember when we fed the starter chicken feed supplemented waddling dew with, we found significant lower count of bacterial endospores. However, maybe to advocate for further studies that the property, or the mechanism behind this bacterial endospore count need to be explored further, maybe using in vitro studies. We also found that actually feeding both crickets, the two diets, they actually harbored similar bacterial community. So that's my short presentation. I'd like just to acknowledge that this project is actually funded by VRIS. These are partner institutions, KU and JUST, Jaramogi Oginga University in Bordeaux. Thank you very much for listening to me.
Thank you very much, Jeremiah and Tim, for the wonderful presentation and the research you are doing on the line of edible insects. So there's really a lot that can be done on edible insects, be it for feed or for human consumption. So open the floor for some questions. So the first question is, did you add the weed to already commercial feed, or you formulated the feed incorporating the weed? Let me check it again.
Yeah.
Did you add the weed to already commercial feed, like buying commercial feed and you add the weed, or you formulated the feed including the weed?
Okay, what you did, we formulated the feeds, but the weed was actually in fresh wheat basis. We just obtained it from the farm, but the chicken feed we formulated.
Okay, so the weed was incorporated into the formulated feed?
Actually, I would say that I supplied, like, because remember feed is on dry wheat basis, eh?
Okay, all right.
And the weed are in fresh wheat basis, eh? So I would supply, for instance, chicken feed and the waddling dew. I would provide the chicken feed, I would separate, then the waddling dew on fresh wheat, like leaves.
All right, okay. Thank you for that. Maybe any question?